Welcome again to Let's Talk Forensics with Pastor Dan. We're going to take 2 days here to perform two different one lab and one activity about blood stain. Now, we're not going to work with human blood, so you're going to need to make for yourself a blood simulant. Now, there's three things you'll need. The first is Karo syrup, so any generic Karo syrup. Second is water. Okay, so you want to be careful how much water you add. The more water you add, the more loose it'll be, the less water, the more viscous it'll be, the more congealed. Blood seems somewhere between, so add a little bit, test it out. And the last thing you'll need is a little red food coloring. Now, be careful with that as well, because if you add too much, it gets really, really, really dark. If you don't add enough, it looks really fake. So once you make that up, the first lab we're going to do involves this right here. So this is a blood stain lab. So we're going to take the sample we make, go to a specific height. We're starting in meters here because doing science, 0.5 meter, 1 meter, 1.5 meters. But if you want to do feet, you can do that as well. 2 feet, 3 feet, 4 feet. And you're going to drop it. Now, as we said, gravitation will increase in speed until it hits and it'll get bigger size. So we'll be able to identify it. Then we'll also want to try to do some directional. So try to drag it and see if we can produce those things. You want to sketch those out, get dimensions, and then identify these questions. All right, so that's the first lab we're doing. It's going to be a lab on blood stain drops. Then I don't have a lab for it. I just sort of always did it. I would want you to try to identify blood stains found behind. So take your mixtures and then get lots of newspaper, because we want to soak through. Do a pool, so just pour some of it out. Do a gravitational, so do the same thing, drop it out. Do a directional, so have it pulled out. Do a spray, so the way you do sprays, put a little bit into a straw and blow it so that sprays out, and then do a cast off. So I always would take a brush of some type and just fling it on there. Now, you want to have something set up so that you as the investigator can go in and identify. So this is site 1, what kind is this? This is site 2, what kind is this? This is site 3. So 5 sites, identify the different types of splatter that produce. So 2-day activity. First day we're doing the lab, that's right here. Put it up so you can see it all right there. And the second is the activity with the blood splatter. So those are 2 things we're going to do. Set them up. Water, Karo syrup, red food coloring. Hopefully that's not too bad. Take care. God bless.